Welcome to Mr. Chalk's Revision Tips. In this video we will look at the different types of chlorophyll, the accessory or the accessory pigments, and the chromatography of chlorophyll. So in photosynthesis, the sun's energy is used to convert carbon dioxide and water into glucose and oxygen. So to do this, the plant uses chlorophylls. There are a range of different chlorophylls that it uses to carry this out, not just one. It has accessory pigments to help it absorb the light, and we can go and separate these different types of chlorophyll out using chromatography. In photosynthesis, the sun's energy is converted into chemical energy by photosynthetic organisms. However, the various wavelengths in sunlight are not all used equally in photosynthesis. So, instead, photosynthetic organisms contain light-absorbing molecules called pigments that absorb only specific wavelengths. So, the set of wavelengths absorbed by a pigment is its absorption spectrum. And we can see in the diagram that different types of chlorophyll will go and absorb light at different wavelengths. So the set of wavelengths that a pigment doesn't absorb are reflected, and it's the reflected light that we see as the colour, which is why we see plants as being green. There are 5 main types of chlorophylls: chlorophylls A, B, C and D, plus a related molecule found in prokaryotes called bacteriochlorophyll. So in plants, chlorophyll A and chlorophyll B are the main photosynthetic pigments. Chlorophyll molecules absorb blue and red wavelengths as shown by the peaks on the previous absorption spectrum. Structurally, chlorophyll molecules include a hydrophobic tail that inserts itself into the thylakoid membrane and a porphyrin ring head, so a circular group, that absorbs the light. So because of the central role of chlorophyll in photosynthesis, all pigments used in addition to chlorophyll are known as accessory pigments, including orochlorophylls as well as carotenoids. So the accessory pigments allow the broad range of wavelengths also to be absorbed and thus more energy to be captured from the sunlight. Carotenoids are a key group of pigments that absorb violet and blue-green light. They're brightly coloured, so that's why fruits and such forth look bright. So in photosynthesis, carotenoids help capture light. They also have an important role in getting rid of excess light energy. So when the leaf is exposed to full sun, it receives a huge amount of energy. If that energy was not handled properly, it could damage the photosynthetic machinery inside of the chloroplasts. So carotenoids in chloroplasts help absorb the excess energy and disperse it as heat. Accessory pigments are light-absorbing compounds found in photosynthetic organisms, like we've just said. So chlorophyll B transmits green light and mainly absorbs blue and red light. Xanthophyll pigments pass along the light energy to the chlorophyll and act as antioxidants. The molecule structure gives xanthophyll the ability to accept or donate electrons. Anthracene pigments absorb blue-green light and aid chlorophyll A. So apples and autumn leaves owe their vibrant reddish colour to these compounds. Chromatography is the process of separating different chemicals, compounds. So chlorophyll is a pigment that absorbs sunlight and is made up of a range of different chemicals. So if we look at how we go and we carry this out. So we start off by getting a sample of leaves, crushing them up and covering them with a small amount of propanone. You grind the leaves thoroughly into a pulp as this increases the surface area and allows the propanone to get physically at the chlorophyll. After you've done that, it's important to examine the liquid. So if there's no free liquid, just add a little bit more propanone. If the liquid is not dark green, then add some more leaves. Then we can look at filtering it. So we go, funnel up a piece of filter paper and put it into a test tube. Pour the greenish liquid through. So this will separate out the chlorophyll that is dissolved in the propanone and leave any other cellular material or material from the leaf behind. We can then start thinking about doing the chromatography. So we draw a pencil line across a layer of silica gel chromatography paper about 1 centimetre from the end. Then use a micropipette to put on a small drop onto your TLC plate. Allow the spot to dry, then add another drop. Carry on repeating this until you have a really concentrated drop, but is no larger than about 2 millimeters in diameter. Then put your T-cell plate into a chromatography tank in the fume cupboard until the spot at the bottom, with the spot at the bottom. Use your solvent, so the solvent level should not reach up as far as the spot itself. Cover the tank with a black polythene bag. This is to make sure that the light isn't going to affect the chlorophyll. So lift the bag up every 2 minutes just to check that the solvent front hasn't gone too far up the paper, because you don't want it going off the end. So when the solvent front is about 2 millimeters-ish from the top of the plate, remove the chromatogram and mark the position of the solvent front using the pencil. Mark the position of each coloured pigment with a pencil. You can then use this or transfer this either by taking a photo of it or by drawing a diagram to show where things are. Right, because the colours fade quite rapidly. Then measure the distance of the solvent front and the distance that each pigment has moved. We can then look at calculating the RF value for each pigment, and by that we can go and use a table to see what the pigments actually are. So to calculate the RF value, we divide the distance moved by the compound dissolved by the distance moved by the solvent. We can then go and use a table to go and work out what the individual drops actually are.
Thanks for watching.